It's my greatest pleasure to introduce Professor Marilu Gorno Tempini. I have worked with her for 7 years when I was at UCSF, so I'm particularly thrilled and honored to have her, sort of my big sister, speak today as a speaker. She is a Charles Schwab Distinguished Professor of Neurology and Psychiatry and Behavioral Sciences at University of California, San Francisco. She directs the ALBA Lab at the Memory and Aging Center. She co-founded and co-directs the Dyslexia Center and co-founded and co-directs the UCSF UC Berkeley Schwab Dyslexia and Cognitive Diversity Center. Yes, she is a very busy person. She received an MD from the University of Brescia, Italy. I apologize if the pronunciation is not correct. And then received her PhD in imaging neuroscience in a record time in 3 years at the Institute of Neurology of the University College of London with Kathy Price. Her clinical work concentrates on behavioral neurology across the lifespan, and her research investigates a neurobasis of higher cognitive functions such as language and memory. Marilu has applied her expertise to neurodegenerative diseases, particularly primary progressive aphasia and frontotemporal dementia, and to language-based learning differences such as dyslexia, and which we hope she'll talk about many of these things today. She's an exceptionally prolific researcher, as you know, with over 230 peer-reviewed publications alone, and at least 8 major NIH and other grants where she is PI. And if you see her CV, you will see not only her prolific career, but also her care for science and her mentees. She has mentored a number of now well-known faculty in many of her fields, such as Bill Seeley from UCSF and Stephen Wilson at Vanderbilt University, to name a few, with a total of over 70 students, postdocs, and junior faculty. I'm always so impressed by her scientific curiosity when I attend meetings with her, eagerness to learn, and her methodical and incisive approach that she takes in research. So with that, it is such a pleasure to have Marilu today as our first speaker to kick off this academic year. Please join me in welcoming Professor Marilu Gorno Tempini. All right, Marilu, would you like to start your screen share? And you're muted. You're still muted, or maybe it's me. You were just fine before. Did you do something with the headsets? Do you need to put them back in, or? Okay, if other people can hear, please go ahead. It's something with me then. I apologize.
So I to take you through this, finding this link between typical neurodegenerative diseases of older age and neurodevelopmental differences, and how we think this has inspired us to really take a lifespan approach on kind of taking what we've learned in adults and apply it to children, and learn about neurodevelopment from a neurobiological point of view might actually help us treat neurodegenerative diseases. So language, it's a model that we use. Mainly, I'm interested in, the lab is interested in the neural basis of higher cognitive functions and human behavior. We use language as a model because we know more about language, and so it was an easy path, and we, of course, really interested in it. But as we know, really the study of a patient born Broca's first patient was the beginning of behavioral neurology and cognitive neuroscience. So he was really the first one, and thinking about it, it wasn't that long ago, to really link a lesion in the brain with an altered behavior in a human subject. So what he observed is that this patient that he saw in the clinic had a lesion in the third frontal convolution and had trouble speaking. And of course, there was no imaging at that point. So the only way he actually had was to then do a pathological study, the same investigator clinicians doing both, which we went back to a UCSF, and we'll talk about it a little bit. So the bias really since Broca for many decades, a century, I would say, was that to study language in patients, we would use as model stroke. And that implied that we were basically looking at regions of the brain that are susceptible to stroke. And we made the inference that language is a perisylvian function. And I will argue that basically we made that inference because we only study stroke for a long time, and studying other disorders comprising, progressive aphasia, so atypical focal neurodegenerative disorders and neurodevelopmental disorders, actually helped us have a broader, more distributed network view of the neural basis of language. So my first, the disease of interest disorders of interest that we started working on after, as Fumiko said, I didn't quite do my PhD in 3 years, Fumiko. I was, the residency is very long in Italy, and they let you take a year off for research. So I kind of combined the two. I wish.
Got it. It looked impressive regardless.
So I wanted to apply the knowledge that I learned from Kathy Price and the really strict and amazing cognitive neuropsychology world in England to a disease, being a physician. And so I came to UCSF to do a 2-year postdoc, and I'm still here after 20 years. But anyway, I came to UCSF to do a postdoc to study primary progressive aphasia. So this disorder was defined first by Marcel Mesulam at the beginning of the 80s, although maybe the first case from PICS a few decades, a century actually earlier, it was probably a primary progressive aphasia. But in the modern neurology world, primary progressive aphasia was defined by Marcel as a clinical syndrome characterized by basically focal language impairment, word finding, naming, and comprehension deficits that was isolated for 2 years. This was kind of a revolution in the neurodegenerative world at this point, and it was based on this amazing clinical observation. So at that point, neurodegenerative diseases were only Alzheimer's disease, and they only started with memory disorders. That was the state of the art. So for Marcel to write this paper and say, no, listen, an aphasia can be an isolated aphasia, can be neurodegenerative in nature. It can start slowly, not like a stroke abruptly. And it was a real revolution. Now, he didn't have much imaging available either. So based on what we knew about language in stroke, he assumed that the atrophy would be mainly perisylvian, and the neuropathology would be non-Alzheimer's, because Alzheimer's disease caused memory impairment. That was the dogma at the moment. And we'll see how the field evolved from this great observation. At the same time, there were 2 parallel worlds, and I had the fortune of studying in Europe and then moving to the US. So in England, at about this, same time, a little later, semantic dementia was described. And also non-fluent and semantic dementia was described, and sometimes called progressive fluent aphasia. So the relationship between the two, the PPA in the US and the semantic dementia on the English side of the ocean of the study of the field was not quite clear, but it was clear that they were both neurodegenerative disorders of focal nature. A little later, Murray Grossman described another presentation of progressive aphasia that he called non-fluent. So when I kind of entered the field, there was this kind of a little bit of confusion, and the progressive non-fluent aphasia and the semantic dementia were non-Alzheimer's in nature. That's what it was thought. Although many patients with non-fluent aphasia were thought to have 35% to have Alzheimer's disease and not FTD pathology. And so it was a little bit confused. So I arrived at the Memory and Aging Center, and with the team there, we started taking a systematic approach to this. We took all of the patients that had a diagnosis of general PPA as described by Marcel, and did a voxel-based morphometry analysis and saw that there was perisylvian atrophy, but also these anterior temporal atrophy that had been described in semantic dementia. And then we took a cognitive neuroscience approach and really did a systematic evaluation that we could call phenotyping, clinical phenotyping of the different functions that we thought these different brain regions observed. And we were able to identify 3 main variants of the progressive aphasias. And we'll go through them today, and I'll try to, at the end, there are 3 separate diseases. So I'll try to go as quickly as I can, but please do interrupt me if you have any questions or observations. I'm very happy to make this a conversation. So 3 main brain networks and 3 main clinical variants. It took a while to get this framework accepted, and it is in no way comprehensive. There are more variants than just these 3, but I think this gave us a good starting point to then study more rare presentations. And what is really unique about studying these disorders is that these 3 variants have different pathology in the brain. That means they're caused by different neurodegenerative diseases. So diagnosing them is not just important for us to understand better the brain or to exercise our physiology skills, which with stroke it became from a clinical point of view a little bit more, because we have imaging that can see lesions, stroke lesions or vascular lesions. Here it was harder to predict pathology, like molecular pathology, from just looking at the scan. So defining these clinical syndromes really precisely had a very important diagnostic value for the patients if we're studying therapeutical interventions, pharmacological interventions that are hopefully very close. So the logopenic variant, the one that has temporal involvement and is mainly a disease of phonology, is mainly caused by Alzheimer's disease. And these are young patients, usually in their 50s and 60s. So these are the patients that we want in clinical trials for Alzheimer's disease. The non-fluent agrammatic motor speech variant is mainly a tauopathy in the brain. So that's what the protein, the toxic protein that deposits in the neuron in the brain and causes neuronal death is a tau. It's a different disorder. And the semantic variant or semantic dementia is again another disorder, TDPC pathology. So these 2 are in the FTD family. This one is Alzheimer's disease. So it's never complete. As you can see, in no case is 100% because the anatomy, the clinical presentation, the aphasia and the motor speech disorder and the anatomy always correspond if one is experienced enough clinician. From the anatomy to the molecular toxic protein pathology is only a probability diagnosis. But this is to give you, it's probably a lot at the beginning, and we'll go through this a little bit more in detail, but just to give you a sense of how important this is, the differential diagnosis between these 3 families of aphasias. So they're also an incredible model to study vulnerability, network vulnerability in the brain. And this is a concept that I'm really interested in applying to neurodevelopmental disorders, and we'll get to that. But one of the main discoveries in neurodegenerative diseases in the last, I would say 2 or 3 decades, is that really each disorder that we talked about, Alzheimer's, TDP, tau, are prefer certain networks in the brain. These networks in the brain, for reasons that we don't know yet, that are actively being researched in many labs around the world, are more susceptible to this specific disease. And the disease, the toxic protein, then spreads transsynaptically. So it gives us a great model from both the clinical and the neuroscience perspective to study network level dysfunction in the brain. So for some reason that we will maybe go through some of it, there's certain epicenters and nodes in the brain that have intrinsic vulnerability to a disorder. So it can be genetic, and we'll see maybe developmental. It can be environmental. It could have to do with lifetime brain usage and reserve. So this is just a scheme, of course. So we can think in hypothetical disease hitting the brain and first hitting the epicenter that has greater vulnerability to that disorder. And then it would spread, the hypothesis was, in a predictable way based on the connectivity of that epicenter with the rest of the nodes in the brain. So from epicenter 1, you might hypothesize that it would go to node 4 and then spread to the rest of the network. So we will take this model and try to apply it using imaging and cognition and the amazing cohort of longitudinal patients that we've collected in 20 years, with data going all the way from detailed speech and language evaluations all the way to the molecule and pathological diagnosis at the UCSF Brain Bank. So, fortunately, I have brilliant bioengineers and neuroimagers in our group that helped us think this through. So this is the work of Giovanni Battistella in the lab. So our first step was to really create intrinsic language connectivity networks that are related to the PPA networks in healthy individuals. And we chose these main epicenters to really characterize in healthy controls the networks that we think are most susceptible to our diseases of interest. And using all the literature that we have from the 2 centuries of study of language, we can make some prediction regarding what these networks actually do. So it was quite striking after, having struggled doing activation fMRI studies with patients who cannot do tasks for a couple of decades, what we could get in 10 minutes of resting state fMRI. So we could basically map these networks that cover most of the left hemisphere. You would notice, and we had many discussions with my close collaborator and friend, Nina Drunkers, that we kind of missing a Wernicke's area in these disorders, in these networks, and in our disorders, really, because that is really not a hub in this neurodegenerative diseases, an epicenter, which again puts a very a different emphasis of the importance of certain other epicenters and hubs in the network compared to stroke aphasia. As we know, there are specific white matter tracks that connect these epicenters and hubs, so we'll look at those as well. And so let's go into the first variant. So this is a non-fluent agrammatic variant of PPA, hub in the posterior or premotor region of the inferior frontal gyrus. Many patients for very long, and still now, when they have a neuroradiology, a clinical MRI scan, they often come to us with scans that are read as normal. So if any is interested, you can see, always look at the asymmetry. So atrophy, I mainly use neurological conventions. So right is right and left is left. I showed you a case that is quite visible, but many times clinical scans are read as normal for age. The main characteristics is motor speech planning and execution difficulties, apraxia of speech and or dysarthria with speech hesitant, effortful, and sound distortions, and or agrammatism. This is a big discussion in the field. Do we want to have separated versions that is mainly motor speech and mainly agrammatism? That's totally fine. The difference doesn't predict pathology, but it is certainly true that there are patients that present with a progressive motor speech deficit, and rarely as a isolated agrammatism. Most patients have both, and they have spared semantics and comprehension. So I'll show you an example. This is a classic picture from the Western aphasia battery. So I always think that seeing a patient kind of makes everything more real and relevant and important.
Oh, a man, a woman have a picnicky, a boy flies a kite, and next to the boy is a dog. Maybe they are family.
So this gives you an idea, and maybe I didn't emphasize enough how focal and mild the speech and language impairments are in these patients at the beginning. I like the term primary progressive aphasia because it doesn't have the dementia word in it. These patients for many years are not demented. They have a progressive language impairment. They have an aphasia. I mean, they present as aphasic. The cognitive mechanism behind the communication problem might not be a language impairment, but they present clinically as a language impairment. Their memory is spared, their visual spatial function is spared. They're still working when we see them. They're driving the car. Of course, unfortunately, they're neurodegenerative disease. They get worse over time. And so people eventually become demented, but I've always had as a clinician, kind of dislike of using the term semantic dementia, for instance, for that term, because at the beginning, they're really not demented. So we briefly said that the epicenter of this disorder this is a VBM of a patient, of a group of patients with non-fluent PPA, is in the posterior part of the IFG, premotor or percular regions. This is the healthy network in controls. This is the work of Maria Luisa Mandelli. So this is healthy network in controls, and the Aslan tract that connects the IFG to supplementary motor area and pre-SMA and basal ganglia. So this is the network that is linked to this epicenter of disease in healthy controls. So our hypothesis would be that if in early patients where the disease starts in the inferior frontal lobe, it would spread to these other regions, making the motor speech and language impairment more severe, especially language impairment when it goes more anteriorly, and motor speech more severe when it evolves into SMA, pre-SMA, and basal ganglia. And the longitudinal change would follow a spread of the tau protein within mainly the Aslan tract of the left hemisphere. Unfortunately, what we know about these diseases is that the non-fluent PPA is that most of the time it starts as an isolated language impairment, and it becomes a more severe generalized motor impairment, such as cortical basal degeneration of progressive supranuclear palsy. So for the non-clinician, a general Parkinsonian syndrome. So does intrinsic connectivity network anatomy and topology in healthy controls predict the atrophy in the patients? This is our first step. I don't have time to go through all the methodological details, but this is again work that Giovanni did, showing that with an unbiased data-driven classification approach, it is, oh, this is not advancing here, let's try here. We do identify in about 150 patients, 3 clusters. And the first cluster corresponds to patients who have atrophy in these dorsal speech and language network that corresponds to the atrophy in the non-fluent PPA patients. And Maria Luisa work actually put together this with our longitudinal changes in patients. So we see again this network in healthy controls, and this is longitudinal change in our patients at a year, 1 year apart, so year 1 and a year later. And she showed that indeed the network that was most strongly connected was the one in which there was greater change longitudinally. So really, if we take this network-based approach, we can predict where the atrophy will go after a year, which has huge implication both clinically and also for providing outcomes to clinical trials. So we have the fortune of having this incredible multidisciplinary collaborative group at UCSF and have a brain bank. This was actually my first patient that I saw with non-fluent PPA back in 2001. And it was one of the experiences of my life to have her brain in my hands, thanks to Bill Seeley and his brain bank. So you can see in the macro view of the brain, the thinning and atrophy of the posterior portion of Broca's area and premotor cortex. This was really one of our first autopsies, so we didn't take a picture of the medial view, but you can kind of see a dip here that's probably due to atrophy in the medial SMA, pre-SMA regions. As I was saying, pathologically, the non-fluent PPA group is disease is mainly caused by cortical tauopathy, and in most cases, a cortical basal degeneration. These are kind of details, but what you can, in summary, this is different from Alzheimer's disease. It's a frontotemporal dementia spectrum pathological disorder in which there are deposits in the brain of particular repeats of tau. This is a 4-hour tauopathy. It involves white matter a lot, much more than Alzheimer's disease. So it kind of gives us a model to study white matter connectivity as well. Some cases, the ones that have more motor speech deficits and often more dysarthria, this goes to a little bit more if anybody's more into this field. It's probably predictive of the progressive supranuclear palsy. Can hear a couple of seconds of this patient, you hear the characteristic of her speech is quite different.
In the foreground, the man and the woman and
so more dysarthric, less speech planning and more speech execution. And in this case, this patient had a progressive supranuclear palsy. And I wanted to show you her brain to show you the striking deposits of, you can almost see the tau protein travel within the axons, right? You can imagine this is the IFG epicenter. You see it in the this is where the sample is taken. And you can see the tau spreading through the axons. And basically this would be the beginning of the Aslan track. Okay, this would be a good time if anybody wants to ask questions regarding this variant. Is really, again, as I said, it was born as all one disorder, but they're really 3 different disorders.
Great, thank you, Marilu. There's a question from Jill first, and then we'll go to William Graves. But Jill writes, cries right here. Hi, probably I missed it, but how many subjects were necessary for the data-driven classification? You said you were going to skip through a little bit,
Yeah.
and what kind of classification model was used?
So it was, this is data collected through 20 years, right? We basically see about 10, 15 patients a year with each variant. So the ones who have longitudinal imaging and pathological diagnosis, we basically have a cohort of about 150 non-fluent progressive aphasias. Exactly which model Giovanni used, I don't know if Giovanni's here, or Malu, maybe you're here. You can speculate a little bit more on that. I know it was a similarity matrix, but...
Hi, this is Giovanni.
Hi, Giovanni.
Yeah, just it's based on a similarity matrix. Basically, the slide that Marilu showed about the resting state networks and the atrophy map that we created in our patient based on gray matter volume, what we call the W map. So basically, an individualized atrophy map. So we just created a similarity matrix between the 2 measures, and then showed in the matrix how the 3 variants, basically the correlation of subject in the 3 variants.
Was there a publication there that you can cite and I can type it in the chat box, or is it?
Yes, there is a Journal of Neuroscience publication. I can put it on the chat.
Oh, great, thank you.
Thank you.
And
Marilu, we can...
there were about 300 patients total in that matrix, like something like that?
I don't remember. I think it was probably close to 200. But
100, okay.
I saw number 230 on the slide, I think, but I'm not 100% sure.
it
Great.
might be. It might be.
Thank you for typing in the reference. William Graves?
Yeah, just a quick clarification question. It's a beautiful talk so far. The last slide you just showed with the pathology, is the tau or the tau deposits the dark brown stuff or the blue stuff?
The dark brown stuff.
Great, thank you.
They're depositions, like these are like, they're not plaques, and I'm blanking on the name now, but these are actually these, when you look at the brain, they're really like little rocks, deposits of calcified tau. And then these are, the brown filaments are in the neuron.
Great,
So,
thank you.
yeah.
Great, thank you. I think that's it for now. So if you can move on, that'd be great.
Great. So anterior temporal lobes, these are really my passion. I think they're super interesting. I think the field of neurodegeneration here really opened a whole new chapter in the science and clinical care of the language disorders. So this area, the anterior temporal lobes, are not really hit by stroke in isolation. Very rarely. It's not true that it's never, but rarely, because they basically are irradiated, like they get blood from both the carotid artery and the vertebral artery. So both the anterior and the posterior circulation in the brain. So when one fails, the other one takes over. I think because it's the most important area of the brain. So, you know, it's the area that does the Platonic idea of our concepts. So it's involved in long-term memory and of our concept. We also know as neuroimagers that it's a very difficult area to image with fMRI. There's a lot of distortions, and we've tried in many different ways to recover signal from there, and we're much better at it, but it still does not have the same signal to noise ratio as other regions in the brain. So it's been ignored for a long time in our fMRI, activation fMRI experiments. And it's also difficult to reach with TMS and with MEG. So I think these patients and also patients with focal epilepsy have been really the models to study the functions of this area of the brain, that it kind of has, this is from a beautiful review from Marcel Mesolom again. It's kind of a mix of different type of cortex, you know, very new association cortex, neocortical region, the cortex, like this pink, and then association cortex, visual and auditory, and then also paleocortex, which is the cortex that is connected to the limbic and paralimbic system. So also involved in emotional reactivity and understanding. So a really interesting region of the brain. So it's involved in semantic memory. This is a very simple scheme for pardon the simplicity, but if we think of semantic memory as part of long-term declarative memory and compare it to episodic memory, what semantic memory is really our encyclopedia, like knowing who Obama is. It doesn't matter how young and happy or older and unhappy he is, still we have our concept. We don't quite remember when we first learned about him, or so the concept is, and even more so for a telephone, the concept is not linked to a specific date and time, to a specific event, and it usually doesn't have an emotional context. Although for faces, we'll talk about that, it's less true than for tools and objects, that there is an emotional content on biographical information about people. But really in contrast with the autobiographical and episodic natures of the events. where we clearly can put ourselves in time and place, and we can remember the emotion that we felt in that moment. That is the episodic memories system. So the two really dissociate in our patients, not completely. Dissociation is never 100%. But the interesting part of semantic memory is that it's usually multimodal. So it's not just knowing the word that corresponds to the concept of telephone is really a semantic feature of the telephone, just like the visual features. And so in the different aspect of these patients with semantic dementia is that they don't just lose the words that are associated with these objects, but they really lose the meaning, what they're for, how you manipulate them, where do you find them, who uses them. In case of animals, where are they geographically? It's not a all or none concept. It's kind of a web of semantic features that they slowly lose. And the features that they lose first, it depends on the anatomy in the disease and on the frequency and familiarity of concepts. So this is kind of the definition from our from the field of semantic variant or semantic dementia. Focal atrophy in the anterior temporal lobe, usually asymmetric, either more in these cases of semantic PPAs, mainly left, although is usually bilateral, but much more severe in one side than the other. Again, these were diagnosed as trauma or stroke by neuroradiology for a long time. I think fortunately the field has evolved now, but do not be discouraged if you think that a patient has this and the imaging read comes back non-consistent. You always have to look at the scan yourself. Focal, so frontal and parietal lobes are usually spared. Clinically is a striking clinical syndrome, and I'll have 2 or 3 videos to show you, but they have a very severe confrontation naming impairment because, again, the name, and especially a proper name, is is one of the most labile semantic features linked to a concept. They have trouble understanding words. They have trouble recognizing, identifying object, famous people, sounds that correspond to object. And they have an interesting deficits in reading and writing and orthography that is called surface dyslexia. So they have trouble reading irregular words, but they can decode pseudo words, kind of the opposite of developmental dyslexia. Later in the disease, and we'll see why, they can develop behavioral changes. They have strikingly spared phonology and grammar. And this is very different from patients with Wernicke's aphasia. I'll always remember Nina's drunkard's reaction seeing the first semantic dementia patient with me, and she was struck because you would never see such a severe naming and word comprehension impairment in a patient that is so fluent and grammatical in a Wernicke's aphasia. So here are some examples of a patient naming items. You'll notice that the main error that they make is that they generalize. They give supraordinate or ordinate responses. So a semantic dementia patient will never call a rabbit a frog, because those are low frequency, low familiarity items. What they'll say is either animal or dog. They'll go back to the prototypical response because they lose all the specific features, but they'll keep the general instead of dog, they'll say animal. And so here's an example.
Is that a vegetable, a fruit? Fruit. Dog?
Sorry, one last thing. Only for the very low frequency items, they'll actually say, I don't know, because there is no prototypicality. And so they will have a trouble really knowing what the object is. And this was an issue that was very debated at the beginning. That was a lot of confusion when in some papers, a semantic dementia, semantic variant was called a fluent aphasia. And because if you only use like the stroke card items in these patients, they're all mainly high frequency. Stroke aphasia is less sensitive to familiarity than semantic variant is. So the patients will know what the item is because it's a high frequency item. And so then the researchers or clinicians will say, well, that's not a semantic memory impairment because they know what the object is. But that was just because they were using high frequency items.
I don't know what that is. Cat? Fruit? Yeah, I know what that is. It's a, I forget what you call it.
Okay, so you hear how fluent and how grammatical the patient is. And they have this behavior that when you see it once, you'll never forget it. And you hear a comment here. Sometimes you tell them a word and they just have this surprise. They really don't know what the word is. They'll repeat it perfectly. And but they really don't know the meaning.
Over, and if you need flowers for somebody or something for presents, come on over and I'll give. Okay, now what's this?
Take a look at that picture and tell me what you see and try and talk in sentences.
Try to talk what?
In sentences.
What are sentences? See how stupid I am.
Just to talk.
They say, oh, you're not stupid, but I am stupid. I don't know what you're talking about. And
so this kind of sense of surprise, of really not knowing, should know, sounds familiar, I can repeat it, but I really don't know what the meaning is. You can see a hint in our patient there who was a very proper kind of country club, South Bay woman who started being much more familiar, a little bit disinhibited. And this is what sometimes happens in these patients because of the connectivity of the anterior temporal lobe with the orbital frontal cortex through the onset fasciculus. So longitudinally, we would expect these patients to develop symptoms of the right side. So if they are start with the left hemisphere atrophy, it will become bilateral as time goes by, and it will spread to the orbital frontal cortex, causing behavioral changes. And I'll show you an example here.
The thing I'd like you to do is raise your hand.
okay. You want me to get?
That's good. And shut your eyes.
Eyes.
Good. And
so you might say that some of this behavior can be normal in some societies, maybe in Italy, but here was definitely inappropriate. So we were interested, and this is again Giovanni's work with the rest of the group. We were interested in looking at this incredible dissociation in semantic patients. So dissociation between really the ventral language system and the dorsal language system. So we have such a striking deficit in understanding and comprehending single words, not sentences. Their grammar is really good, but single words, and the striking preservation of fluency. So we looked again at our semantic network with the epicenter in the anterior part of the middle temporal gyrus, connecting to the angular gyrus. We know this is part of the default network that we think is heavily involved in semantic processing, and looked at connectivity, intrinsic connectivity at rest of this epicenter, and showed that there was dysfunction, decreased connectivity with the angular gyrus in the semantic dementia patients, although this area was really not atrophy. So this is the networking controls again. This is the pattern of atrophy in the semantic patients that we considered in this study. And we looked at connectivity from this epicenter. As you can see, we chose this and not really the anterior temporal lobe, because as you saw in the previous slide, that part of the brain is basically gone. So we couldn't really put the epicenter there to study the patient's disease. So decreased connectivity with the angular gyrus, despite the fact that there was no atrophy. And then we actually saw increased connectivity in the dorsal speech production network. So still increased connectivity with this area that is more dorsal angular gyrus spreading to the supramarginal gyrus. And there is actually increased connectivity, probably sustaining the fluency sparing in these patients. And actually the strength of connectivity correlates with increased articulation rate in this patient, with articulations rate in these patients. So just one quick word about the right temporal variant. So the field of the semantic, progressive semantic impairment studying patients with the left variant that present as an aphasia. We know the cognitive mechanism is a semantic memory impairment that presents as an aphasia. The patients who present early with only right temporal are super interesting, and really we think they have, the studies are just being done now, but we think they have an impairment for semantic memory that is related to socially relevant items. So they have trouble recognizing famous people early on, and they have trouble recognizing emotional faces. So they present as people who have lack of empathy. So many of these patients didn't get diagnosed for a long time because we would see couples going to couples therapy and not being referred to a neurologist. This is kind of a bias in the field that we can go back to also neurodevelopmental disorders. You know, it's the same disease, but if it manifests as a lack of empathy, it goes to psychiatry and it goes through a whole different, or psychology, and it goes through a whole different route of clinical care and research. But if it presents with word finding problem, then it goes to neurology, and that's a whole different world with a lot of opportunity missed for our patients. So pathologically, we go back, this is a separate disease, very different, although it presents with a progressive language impairment, is called by TDPC pathology. We're not going into details about that, but you can see how, again, focal the atrophy of the anterior temporal pole is. Now, mind you that this is the same patient. You can see that there is more atrophy in the scan than there is in the macroscopic image of the brain. And this is because, unfortunately, near death, there is swallowing of the brain due to lack of oxygen and blood aeration. So there is a swelling in the brain. So you see less atrophy actually in the postmortem macroscopic image of the brain than in the MRI scan at closely the same time. And this is also a patient who unfortunately died very early because he developed a behavioral compunction for drinking alcohol and unfortunately died of acute pancreatitis. So for the clinicians there, always be, you know, these are rare disorders, maybe you haven't seen them, but if you see someone who changes personality or loses empathy in their 50s and they've never had psychiatric disorders before, beware that it might not be a midlife crisis. So the logopenic variant, am I going super slow? Do I have time for the logopenic variant? Super quickly. So the logopenic variant is a disorder of phonological processing. This is, again, our networks in healthy controls. And this is the pattern of atrophy in our first group of logopenic patients. This is kind of a more modern view in cortical thickness. The scans are sometimes even less focal. So you can see, again, asymmetry of volume between the left and right temporoparietal junction. The clinical picture is really word finding problems again. So we go back to the original definition of mesolam. They all have word finding problems, but for very different reasons. Word finding problems because of motor speech issues. So it's not really word finding problems, it's slowness in coming up with a motor planning in the non-fluent. In the semantic patients, is they're losing their semantic features, so they lose words. This is semantic impairment for word finding problems. In this case, word finding problems because of a phonological retrieval and phonological assembly issues. So they have also phonological errors in speech, naming and reading. Sometimes clinically distinguishing a phonological error from a apraxia speech error is not easy. So this was the confusion back then, with having so many difficulties differentiating the non-fluent and this logopenic variant come from the expertise that we need from a speech pathologist and to really differentiate this type of error. As a little side for the young fellows of postdocs in there. I'll always remember after 3 years in an imaging lab, Bruce Miller, who's the head of the Memory and Aging Center, taking me to see my first patient, aphasic patient in England, in San Francisco, and asking me, so Marilu, what do you think? Does this patient have apraxia speech? And being completely puzzled, and because the concept also of apraxia speech didn't exist in Europe, really, a lot of cultural differences. But don't be discouraged if you're not a native speaker to study patients with language disorder. Just record them all. And that's what I did. I would record them and then listen to it and score their errors after, which is proven very useful because now we have all these recordings that we can send through automated speech and NLP pipelines, which will be the future of identifying these errors more objectively. So phonological impairment versus a motor speech impairment, impaired repetition, impaired auditory short-term memory, or phonological loop impairment. So they have trouble repeating sentences. This was really Nina Dronker's observation that really helped define this variant. And the atrophy is left temporal. Without voxel-based morphometry, let's say without computational neuroanatomy, it was very hard to distinguish. You really needed expert eyes to distinguish the mild atrophy in the left frontal versus left parietal. So this is an example, very young patients, and we said these patients have Alzheimer's disease. This is not the picture of the idea that we have of someone with Alzheimer's disease.
Bed.
Bed.
Nose.
Nose.
Pipe.
Pipe.
Window.
Window.
Banana.
Banana.
Snowball.
Snowball.
The telephone is ringing.
The telephone is ringing.
He is not coming back.
He is not coming back.
The pastry cook was elated.
The pastry cook was elated.
Okay, good.
So again, mild difficulties, mainly with long sentences that are not very meaningful. This is a cortical thickness study from Slađana Lukic, who just left UCSF to join you on the East Coast, and a beautiful work showing that the patient's deficits on repeating long sentences really correlated with atrophy in this temporoparietal junction region. So these patients have Alzheimer's disease. So why in the world do they have Alzheimer's disease? Why does this show up as Alzheimer's disease? Have a very long course. We now have biomarkers to diagnose Alzheimer's disease. So it got a little easier for us in the last 10 years. So amyloid PET or tau PET can show deposition of the two proteins that are typical of Alzheimer's disease. I know this is confusing because it's tau like in the non-fluent, but this is 3R tau and not 4R tau. So it's a different type of tau. The hallmark of Alzheimer's disease is really amyloid deposition, but amyloid is not focal. It's all over the brain. It doesn't explain the focal clinical picture. What explains the focal clinical picture is the tauopathy. So because Fumiko came to UCSF, because my own daughter was diagnosed with dyslexia, because Zach and Bruce Miller and other clinicians in the MAC started listening, always listen very careful to the stories of these patients, we realized that many of them had a history of a language-based neurodevelopmental disorder. And looking at the anatomy, right? This is again PPA, this is the typical readers network. There is a lot of overlap between the atrophy in the logopenic patients and the dysfunction or making microscopic changes in the brains of dyslexic readers. So this started to become a model of one of those vulnerability factors that we talked about in the beginning. And Zach meticulously went through all of our charts of our patients, and even retrospectively, and even in this age group in which we know that patients are not diagnosed with dyslexia or language-based language disability, right? They're in their 60s, so they had not been officially diagnosed most of the time. So even just doing a retrospective chart review, he could observe that in the logopenic variant, there was a much higher frequency of language-based learning disabilities than in the other PPA variants and than in the general population. So this was one of our patients. And so what is going on? Who started very young with a logopenic phonological disorder. She was a teacher, so she reported that she was dyslexic, and she had a very long disease course, and more than half of it was only language. So much longer disease course than we usually see in these PPA patients by year 10. Unfortunately, they've usually developed a more widespread problem. In her case, it was a very long clinical history. This is her imaging. You see how mild the atrophy was right at the junction. On the FDG PET, which is metabolism, you see changes more easily. And then her PIB was positive. So she had Alzheimer's disease. So having dyslexia influences the phenotype in these disorders. It starts later and is more focal to phonological processing. So what is happening? How do we study this? Is this just a case of decreased reserve? So individuals with dyslexia have a dysfunction for some reason in different functioning of that network in the brain, and thus is weaker. And when a disease starts, it shows symptoms earlier. That could be a hypothesis, and we can look at that with a tau PET now. Or is there really an overlap of the biology and the biology of dyslexia, of neurodevelopmental differences of whatever type, causes biological vulnerability to these disorders? And here we are again taking advantage of the brain bank to look at actually the brain of patients who had died for neurodegenerative disease or donated their brain and reported that they had dyslexia or they had dyslexia in a first degree relative. And this is a first study in which Salvatore Spina and Zach Miller looked at the brains in 3 of our patients, comprising this woman that you see here, and did show dysmorphic neurons in the wrong layers of the brain that were very full of Alzheimer's pathology. So I'm not going to go through what dyslexia is. I think you all know what dyslexia is. A selective impairment in orthography of neurobiological origin with preserved general cognitive abilities and adequate instructions. It does sound a little bit like a progressive developmental language, not progressive, but a developmental focal language disorder. Most often phonological difficulties. Another time, maybe we'll talk about that. I think that sometimes the phonological difficulties in dyslexia are the tip of the iceberg, just like naming in the PPAs. So it's a symptom, but it might be caused actually by auditory processing difficulties or executive processing difficulties. So I believe that there are different variants, different phenotypes also of dyslexia, but phonological problems are good markers. Is incredibly not cared for. There's been a push in the field not to medicalize dyslexia and consider it mild, consider it a school problem. So it's much less studied and funded than autism or ADHD. Strikingly, there is really not enough evidence on which interventions works and which don't for which kid. I was really struck by this, moving from one field to the other. Even in the neurobiology of it and looking at scans of kids with dyslexia, very few clinicians look at scans for kids with dyslexia. We're ready to give a scan when covered by insurance to our 70-year-old, 80-year-old patients with anomia and selective anomia through insurance, but we're not willing in our society to give a scan to a kid who's 13 and is perfectly smart but cannot read. And that is really also a social justice issue and an issue that in less privileged environments can really create a pathway of discrimination and life challenges. So we thought that generally culturally, the dyslexia is an issue of childhood. But Zach's and others' work are really starting to show that actually the brain correlates of dyslexia are still present in people who are over 60. And they can still show some of the difficulties, focal difficulties in phonological processing that they probably showed when they were young. And Fumiko's works with John Gabriellis and others have shown that kids who are resilient, despite the changes in the brain compared that are typical of dyslexia, they might still read at normal typical levels because they compensate. They have frontal lobe executive compensation methods. And what we think is happening, that's probably after school, when kids with dyslexia have survived school, if they have survived school and not dropped off and ended up in bad situations, they compensate. They find a job that they like, they compensate. And then maybe the deficits come back again when the frontal lobe, sadly, as we all over 50 start to see, when executive functioning starts to be less efficient, then some of the symptoms might come back. If they happen to be unlucky to get Alzheimer's disease, it will show up in a different way than it would normally. So at UCSF, we're really trying to take a different approach to dyslexia and kind of create a program that is similar to the ones that we had for neurodegenerative diseases, multidisciplinary, going from genetics and pathology all the way to classrooms, and really look at brain morphology and correlated with detailed clinical phenotyping. If we actually look at the scans of these kids from a neurological behavioral neurology point of view, we do see little things that are not diseases, but that might cause network dysfunction. And I hope that these kind of studies will help us advocate that evaluation of these kids should come from medical insurance. This is an example of a study that Eduardo Caverzazzi's neuroradiologist now group did. And you can see that, for instance, in this case, you can see hypergerification of the middle frontal gyrus in an adult with dyslexia. So you see the difference. This is not enough to cause significant generalized cognitive impairment. This is actually a super talented individual visuospatially. But it was enough to cause a cognitive symptom. Now, a neuroradiologist that is used to looking at brain malformation that cause epilepsy or intellectual disability would say that this scan is normal. And we would all agree that this individual is normal, is not disease, but still that change is linked to their difficulty in behavioral and a lot of suffering they had through their childhood and life, thinking that there was not knowing what was the cause of their difficulties. So Eduardo, together with others in the team, and Jim Barkovich, who is one of the most renowned neuroradiologists in the world, went through 200 scans of our dyslexic children and really found that in 45% of them, there was a detectable difference. Now, these were scans that were not acquired for this reason, they were acquired for a dyslexia study. So I really think that if we did sequences that were more diagnostic, we might see small heterotopias or changes in the brain that might help us understand better the clinical picture and the neurobiology. I won't go through the study, it's too late, but I just wanted to say that we're also taking advantage of the incredible expertise in neuroscience at UCSF to collaborate and look at different aspects of cognition in dyslexia. This is an auditory study done in collaboration with Yulia Oganian and Eddie Chen that Ting Kui in the lab is leading, and really taking the paradigms that they use in the intraoperative recordings and using them to really characterize the network dysfunctions and also spared and enhanced functions in dyslexia. And we did see that the same area that they see involved in this particular auditory task, which is onset rise discrimination, that they see involved in the posterior superior temporal gyrus in their these are electrodes in their patients in intraoperative recording, really show a striking correspondence with the ability that children with dyslexia have doing the exact same task in an adaptive manner. So this is really striking, really preliminary. Thank you, Ting, for sharing this last night. And actually there we can distinguish two groups of patients with dyslexia and whether they do well or not in this task. So in some of these kids, the cause of their phonological impairment might be an auditory processing impairment, but not in all of them. So, and maybe if we start studying them this way, we'll figure out which treatments work better. So thank you for bearing with me. These are 4 different disorders, but I hope I convince you they're all linked, and that if we have a lifespan approach, always ask your dyslexic kids how their parents and grandparents do. And when we see in the aging clinic how grandparents do, we have to ask about their children and their also their grandchildren. And that will really help us understand them better and understand their biology better. So thank you to the ALBA lab, Language Neurobiology Lab, in kind of a dyslexic way became ALBA, which is sunrise in Italian, and to the whole incredible team at the Memory and Aging Center. When I moved from London, we were 10, and now we're more than 300. It's been an incredible journey that I'm very grateful for. Thank you.
Thank you very much, Marilu. I can't believe it's only been an hour and you covered so much ground. I saw people like John Gabriele and Zach, who you mentioned in your talk here. So thanks to everyone for joining today and staying on over an hour. We would love to take some questions. I know Marilu has made some time, and so she can stay on a little bit longer, I believe.
So, hi, this is Jerome Sains. I have a couple of questions, if you can indulge me, Marilu.
Of course. Nice to see you, Jerome.
The first one is a clinical question, and the second one is a scientific question. So for the clinical question, I suspect the answer is not much, but what can you do for the patients?
Well, it's getting better. It's getting better. So first of all, we have very targeted speech and language interventions that have been shown to slow down the disorder, especially, and what we think that might be even more the case if we combine it with stimulation. Some groups have done that. I've been kind of resistant a little bit, but I think if we combine the right behavioral, like network targeted intervention, cognitive intervention with stimulation, we can slow down the progression of the disorders. I've had my first patients go into a trial for a tauopathy basically this week. So I think molecular changes, molecular therapies are really happening. There are genetic forms of these disorders. FTD spectrum disorders are very genetic. So we have that disease model too. So they're actively studying the use of CRISP to change the genetics, kind of like it's been done in Huntington's disease. I think the first step to really get to proper treatments is to understand the different forms of these diseases, right? You can imagine that if we were going, if we were sending a PPA patient to a trial for Alzheimer's disease 20 years ago, we had no idea what the pathology was. We didn't know. And that might have been the part of the reason why some of the trials were so ineffective, is because we didn't have a good way to diagnose the molecular basis. So I'm optimistic that now that we have biomarkers and better clinical understanding, better stratifications of the patients, these new trials that we're doing now will be more successful. But it has been hard. It has been hard, especially when you get to know these patients so well and you follow them over time, and then you see a new patient in the mild form and you know where they're going. It's really tough. But I think you're about to turn the corner.
So thanks very much for that, Marilu. Now for the scientific question. But first, as a prelude, thank you so much for your talk. It was wonderful to see what you've done in the last 20 years. It's really amazing. And I'm very proud of you.
Thank you.
Anyway, not everyone knows.
That means a lot. Thank you.
But anyway, so the scientific question is this. So it was the work you published in the Journal of Neuroscience was really a tour de force. It was really great. And it takes into account functional connectivity, which is wonderful. But it looks like your approach is still functional neuroanatomy rather than looking at the mechanistic aspects of what's going on between the areas. I wonder if you could comment a little bit about what actually is going awry in the communication among the various areas in the network, not just only with functional connectivity, but also with effective connectivity.
Yes, I think that's a field that we haven't explored enough yet. I think both Ting and Giovanni are really exploring it now. There's certain models, for instance, in the surface dyslexia, I think we have a good anatomical model to test some hypotheses of effective connectivity. We haven't done it yet. There's a lot of complications of doing some of these techniques with patients with atrophy, right? So that's why we have such a strong bioengineering group in the team, but we haven't quite gone there yet, but I'm optimistic that we will. Taking into account what we know about the biology. So, you know, synaptic dysfunction and how these diseases evolve over time, you know, how they start as probably, in some cases, in the tau cases, it's probably, they start as a Y matter disease probably. So synaptic activity might be spared to start with, but that's the challenge, is trying, but also the beauty of these models, of these patients that don't have an all or nothing lesion, like it could be in a big stroke, right? They evolve over time. The more we learn about the basic biology, the more I think we can use more sophisticated imaging tools. But
Okay, thanks so much.
would love collaborations. You know, these are all NIH funded projects.
I have a method for you that you
awesome.
Talk about.
Awesome. This is why I was really excited to participate in this seminar. You know, there's tons of data is available. It took so much work to collect this data set. We're totally open to collaborations and sharing for testing any hypothesis that you have.
That's great. Thank you. Any other questions? I'll ask a question if that's okay, Marilu. Thank you very much. I always feel like you have so much more to say and it always gets truncated at 40, 50 minutes. And so I wanted you to talk for a longer period of time. So this was great, even though I think I can listen to you talk for another couple of hours, even though I feel like I know you and know your work for a while. So my question is, now that you're kind of going into more of a lifespan approach, and that's very, very exciting, the ones that you were kind of introducing towards the end, do you see kind of, do you have an eye on for some biological interventions as well, or does any of what's available, what might be possible kind of excite you or interest you? I know there's a developmental kids sort of delicate issue, sensitivity issue there also, but I'm just curious to see what your thoughts are.
Yeah, thank you, Fumiko. I don't quite have a sense right now. I think we know so little of what is the biological, I mean, strikingly so little, or what are the biological bases of neurodevelopmental disorders, focal especially, like dyslexia. There's been such a little research that I'm not totally comfortable yet in taking an approach that would work for all. Maybe biased on the approach that I've had in the PPA world, but trying to first figure out what is really happening, maybe in the different phenotypes, and then think about therapy. And for now, just try to find targeted behavioral interventions. But I'm happy to be convinced otherwise.
Just one more question, if I can. I'm sorry, you had such great success with this kind of approach of detailed studies and prediction based on their pathways and so on, despite it being more functionally targeted and not as effective as the previous Jerome was asking. But I'm curious to hear if you think these kind of approaches or these kids that we see are as salient or kind of unique in their profiles and will show as strong profiles as you've seen and had success in the kind of aging population?
Absolutely. And I think we'll have, Giovanni has some beautiful 7T images that show more focal changes than we would ever see at 3T. So I think the network, both functional and structural network-based approach would be super important. For instance, as you know, there is this whole issue of overlap between different neurodevelopmental disorders. The ADHD and dyslexia, are they comorbid, or is it the same thing that is manifesting in two different ways? I think effective and connectivity would be a great methods to look at that. And I know you've done some beautiful work on that. I just think we need to. I wouldn't stop at thinking that all the kids with dyslexia have phonological impairment. I think they have phonological impairment as a symptom, but it might not always be the cause, the cognitive and biological mechanism behind their problem. This is the hypothesis. It will take a few years, but it might be like in that patient that I showed. He was diagnosed with dyslexia since he was a baby, but he actually had this hypergyrification and decreased connectivity with the middle frontal gyrus. And I'm sure that influenced how he learned phonology. And so his test of phonology might have been impaired, but that was not the cause of it. And so maybe early on, phonology wouldn't have been the best intervention for him. It would have been, something more helpful would have been like a language-based executive functioning intervention. So this is kind of the hypothesis that will take a lot of work and a lot of different expertise to prove right or wrong.
Yeah, great. Thank you very much. Any other questions?